Good afternoon, everybody. So we've been talking about viruses with capsids and nucleic acid genomes this whole course, and today we're going to talk about agents that don't have capsids or don't have genomes even. That's why we call them unusual infectious agents. So some of the things we'll look at today include what is the minimum genome size you need for infectivity. Yes? One base? One base. Wow, that's some pathogen. One base. No. You need one genome, but how long is it? That's the question. Does it have to be, is 100 enough? Is 10, 500, 1,000? The smallest ones we've talked about so far are circoviruses, which are about 1.7 kb long. But can we get smaller? And the other question, can an infectious agent exist without any genome? Can you imagine if viruses didn't have genomes? The first half of this course would not exist, right? We wouldn't have to learn all those pathways. It would be much easier. Well, there are some agents without genomes. So these are the three agents we're going to talk about. Viroids, satellites, and prions or preons, depending on how you want to pronounce it. These don't fit into the standard taxonomy for viruses, so they're originally called subviral agents. So you'll still find that term in the literature, but they're not really subviral agents. Viroids we'll talk about first. These are major plant pathogens. You don't know any human viroids yet. There is one that is sort of a viroid, but not quite. We'll see what that is. Viroids are plant pathogens. Some satellites are associated with human disease. We'll talk about that one. And prions. What do you prefer, prion or prion? I got about 50-50. Prion, protein, infectious protein in prion. Prion. Anyway, prions cause devastating and fatal dementias. We'll talk about those as well today. First, viroids. These are agents that are only made of RNA. They don't have any protein. They don't have any capsid at all. Yet they still go from host to host. And we'll see how. They don't need receptors. There's no capsid to bind to a receptor. And these, again, are plant pathogens. There is a great website called the Subviral RNA Database. And as of yesterday, it had 2,877 viroid sequences in it. So when you sequence a viroid, you put it in this database, which is apparently hosted up in Canada, yeah.
How often are new viroids found?
I don't know. I'm not sure. Probably as often as you look for them. I mean, this number went, that's a good question. This number went up from 1,500 in a couple of years. So so not every day, but people are actively finding them. There are always new ones. So I would say if you looked on a daily basis extensively, you would probably find every day, but people just don't look. They find new diseases emerging, mostly of plant crops, agricultural crops, and then they identify them. First one was in the 70s. Yes?
Is it the same quasi-species phenomenon in viruses? So that you would only have consensus sequence?
I'm going to repeat the questions because I was just reminded today to repeat them. The last lecture, I'm going to repeat the questions. Are there quasi-species in viroids? Anybody want to venture a guess? You can make an educated guess because you have been educated in this course. You don't know what replicates the viroid genome yet. I haven't told you, right? But what if I told you it was host RNA polymerase II? There would be a quasi-species then? Yeah, there wouldn't be as many as a viral polymerase. There would be some. So in fact, these agents do mutate quite a bit, and it's been studied. And so yes, the answer is there are quasi-species, and I'm surprised at the extent of them, given that they're replicated by host polymerases. Yeah. All right, so that was the first one discovered. Potato spindle tuber viroid, 1967. So there was a disease of potatoes, potato spindle tuber, and so people wanted to know what was causing it, so they made extracts of leaves and filtered them, right? And the filtrate passed on the disease, so they figured it's a virus. But when they started to look at this on the electron microscope, all they saw were these long rod-like structures, which turned out to be just RNA with no capsid. So that was a big deal. That's the prototype, and this guy is 359 nucleotides long. It's amazing. I think these are just amazing things. Some of these cause disease and others don't. All right, so they're mainly an issue of crops. They make crops look bad or taste bad or don't grow properly, so they're agriculturally important. Yes?
When you say 359 nucleotides, do they exist just as separate strands, or is there some kind of separation?
Yeah, we'll see that in a minute. Yeah. All right. Yes?
So, are they never found associated with protein, or is it simply that proteins aren't necessary for transcription?
Well, when you purify these from plants and you look at them, they're just naked RNA. Yeah. So the capsid is not needed. I suppose proteins could stick to them, but it's absolutely unnecessary. Yeah. So they're unencapsulated infectious RNAs. So take just the RNA and you put it into a plant cell, it replicates. And it doesn't code for any protein. So not only is there no capsid, but it doesn't code for any protein. Isn't that amazing? So just think, if we just had viroids, the course we didn't have to do translation, we didn't have to study RNA replication. Oh, but life isn't so simple. The RNAs are very small, they're circular and single-stranded circles. So remember the question on the exam? Yeah. So I might have actually said at some point in this course that there are no single-stranded circular RNAs. If you look back, which I know you won't do, but you might find it, and it's wrong because there are these viroids. Not viruses per se, but there are viroids that are circular. And these get mechanically introduced into plants. The farmer has a tractor that's got viroids on its wheels because it's run over viroid-infected plants, and he or she runs over other plants or mechanically introduces them in that way. And so they're now, we don't call them subviral agents, they're put into two different families with wonderful names. Pospiviroideae is the first one. These are the ones that replicate in the nucleus of the plant cell. And then we have Avsunviroideae, which replicates in chloroplasts. I don't know why there are two O's here. You know, it gives it that certain roll on your tongue, but I can't do it. Yes?
You had mentioned when you were saying they were replicatosity when they're mechanically introduced using a tractor. How would it happen without a tractor? Like, how would viroids have been passed from plant to plant without?
Well, if they're in the seed, they're contaminating the seed stock that the farmer uses. They plant seeds, all the new plants will have viroid. Could be that insects transmit some of them. Animals? Yeah, who tills the fields? Animals till the fields? Oh, you pull it like a horse pulling a till behind the... Okay, I got it. But yeah, the horse could break plants occasionally. So, you know, we get it into a few plants, then the question is how does it spread from plant to plant? It's in the sap, so birds or insects could transmit it among them. But they occur in outbreaks, so a whole North 40 will be infected with a certain viroid, and so there has to be a good way to spread it. These Avsunviroideae replicate in chloroplasts, we'll see in a minute. So these are some of my favorite viroids. There is the kadang coconut viroid. They have the best names. Plant pathogens have the best names, you know. And they're abbreviated with a D at the end for viroid, not just V, because you don't want to confuse them with viruses, but viroid. And this causes a lethal disease of coconut palms. Okay, so that's bad, especially if you like pina coladas. Then we have the hop latent viroid, which infects the hop plant, which you use to make beer. But it doesn't cause any problems, so you can still make beer with viroid-infected hop plants. Although it would be interesting to know if viroids are in your beer, right? That would be an interesting study. Anyway, so that's the hop laden. Then we have the apple scar skin viroid, ASSVD, which causes mild symptoms in apples. They look like this. They have a blotchy appearance, and they actually will sell these in markets. And if you don't like the way it looks, you don't buy it, but most people will look at it and go, what a pretty apple, and they'll buy it, and it's fine. And you're probably eating viroids a lot when you eat these things, but they don't infect you as far as we know. Not yet. Not yet. Someday they will. So viroids don't encode proteins or mRNA. They're circular molecules, 250 to 399 nucleotides long. So that's about the lowest, smallest size for an infectious agent, 246 nucleotides. The RNA is extensively base paired. And that's why it looks like a rod when you take pictures of these in an electron microscope. You don't see actual circles of RNA because the RNA is all base paired, 50 nanometer rod. And many of the viroids are what we call ribozymes. Everybody heard about ribozymes before in some other course, right? These are RNAs that can cut themselves. They're called autocatalytic RNAs. And the viroids that have these, some of them do, some of them don't. The ones that do have it, they're essential for replication. If you remove the ribozyme activity genetically, then these guys don't multiply. So it's essential for replication. So these viroids have a very distinct style from viruses. Viruses we call parasites of the translation machinery of the host. The viroids are parasite of the transcription machinery. Of course, they also have to use the translation machinery, but they go one level lower, if you will, on the flow of information. They need to use the host transcriptional machinery. So here's what a typical viroid looks like. Potato spindle tuber viroid, the first one discovered. And you see it is a circular single-stranded RNA. If you could melt this, it would be a circle, but it's base paired extensively throughout its length except for the two ends. And all these viroids have typical regions, which are shown here. There's usually a central conserved region. There is a terminal right end and left end, of course. There's some variable regions, and then there's some regions which, when mutated, affect pathogenesis of the viroid. They don't affect its ability to multiply in the host, but they affect disease symptoms. So most of them look like this. Some are more complicated. They have more secondary structures and so forth. So how do they replicate? Well, these are circular RNAs, and they typically replicate when host RNA polymerase II copies this RNA and makes concatemers, a concatemer being a long RNA with multiple units of the genome in it. And one way this is done is by what's called the rolling circle. If you have a circular RNA or any nucleic acid, a polymerase can start copying it, and then as soon as the polymerase makes a complete copy, then the extra nucleic acid starts to roll off. It's the rolling circle. So polymerization continues on the circle as new genomes are made. So these are joined unit length. They're multimers of the genome. That's why we call them concatemers. And these are cut, for some viroids, by the ribozyme. Okay, so you have to take the multi-unit length genome copies and cut them into unit lengths, and that's done by a ribozyme. And the ribozymes were discovered in 1981. This was a big deal. It got a Nobel Prize because it's an RNA that can catalyze a reaction without a protein. First time that was discovered. And some viroids have what we call the hammerhead ribozyme because it forms a structure, a secondary structure that looks like a hammerhead. This is, again, an autocatalytic self-cleaving RNA. It folds in such a way that it can just cut itself. It's a really neat structure, and it's been very useful also experimentally. If you want to make transcripts that are exactly the right length, you put a ribozyme at the ends, and it will cleave itself. So now you have a very nice transcript. So these ribozymes, as I've said, are used to cleave the multimeric structures that are made during the replication of these molecules. So not all viroids have these ribozymes, some don't, and the ones that don't use host enzymes to cleave the genome. Okay, so it's either a host RNase, an RNA that cleaves it, and this is obviously a specific reaction. It doesn't just chop up the viroid transcript, but it cleaves it very specifically. So these have obviously evolved to do that. So let's look at two examples of the replication of these viroids. These are the Pospviroidae. They replicate in the nucleus, and they have ribozymes. So these viroids enter the cell. They make their way to the nucleus, and then the RNA polymerase II, the RNA that makes mRNAs from your genome, copies the viroid RNA. So the viroids are plus-stranded. The copy is a minus strand, and it's a concatemer. It has multiple units of the genome. We just show 2 here, but it has many more than 2 typically. This is then copied again to form a plus-strand copy, and this is shown here, maybe being done by Paul II. That goes in the nucleus, where then, it goes in the nucleolus, where it's then cleaved by the ribozyme into unit-length genomes. So each of these is now a genome. Those unit lengths have the ability also to ligate themselves into a circle. They have enzymatic activity to do that. So now we have circles. So we've replicated the viroid genome, and these go out and move to the next cell. You know, plants have these pipes that link cells to one another, and the viroids just go through them. Yes? Yeah, RNA polymerase II is a host cell enzyme. It copies DNA and makes RNA from it. So that's what the ribozyme is using here. Okay, those are the Pospviroidae. Then we have the Absunviroidae. Maybe there aren't 2 O's in the name. These guys replicate in chloroplasts, interestingly, which we haven't encountered yet in this course. And these come in cells, as shown as the highly structured viroid gets into the chloroplast, and these are 2 host proteins that are thought to participate in the transcription and processing of this viroid. So there's a host protein called NEP and PARB-BP33. And you can see there are question marks after them because it's not clear yet. But one copies this molecule by a rolling circle mechanism to make concatemers. And of course, to do that, the genome has to be denatured, of course. And then it cleaves them into unit lengths, which refold. And then they're copied again to form plus strands, which make new viroid genomes, which then go on to the next cell. So, you know, plant cells have transport mechanisms between them, and that these viroids go from cell to cell using those specific transport mechanisms. So what you need to remember is that some have ribozymes that cleave the genome, and some don't. And in both cases, the RNA is replicated by cellular enzymes, because these don't encode proteins, remember. So here's another one with a great name, peach latent mosaic viroid. This is a little longer than the potato spindle tuber, and it's a little more structured. But I'm showing you this to give you an idea about the ribozyme sequences. So here are the 2 cut sites for the ribozyme. And of course, in the duplex viroid genome, these are not active. The highly base paired structure, they're not active. They're only active when a transcript is made and there's different structure. And then the ribozymes will be cleaving, and there's a ribozyme on both strands. Yeah?
Well, we were talking about when derived vaccines.
Yes.
The consequence of contamination.
That's a good question. So in a viral vaccine made in plants, would it be contaminated with viroid? Well, presumably you would make it in a plant that is free of viroids. You can do that.
Would it act as an adjuvant?
It might. Yeah, that's a good idea. But I don't think it would be a good idea to give everyone viroids, because you never know. You could select for one that infects. I don't know. I would keep it viroid free and add some other kind of adjuvant. But yes, a nucleic acid would act as an adjuvant, sure. So where do they come from? Anybody know where viroids came from? Don't look at your sheets. I already gave you the answers. I forgot. Viroids came, I thought you might say from other viruses, which would be a good guess, but they have no homology with known viruses, so that's probably not likely. They came from wild plants, and they were transferred to breeding crops. So at some point, wild crops are used to derive breeding crops, and the ones that were used were infected, and it wasn't known. But where they actually came from before that, we don't know. Because obviously they've been in the wild plants for a long time. They may be very old. They may be relics of the RNA world and have been around in plants since then. But anyway, now in agriculture we use genetically identical plants, a practice that's called monoculture. So they all end up having viroids because they're all derived from the same seed stock. And companies will sell breeding lines globally, so every farm that's producing a certain crop globally will also have the viroid as well. And as we said, they're transmitted among the plants by contaminated farm machinery. I guess it could not just be the tractors and the tools, but also irrigation systems could transmit them as well. The interesting question is how they cause disease. So they're agriculturally important. They lower crop yields. They make unattractive fruits or vegetables that can't be eaten, so they're economically important. But how do they do this? And this has only been revealed very recently because it looks like it involves small RNAs, small interfering or microRNAs. The idea is that when these viroids infect the plant, they're chopped up into small microRNAs, 21 to 24 nucleotides long, and these enter the RNA silencing apparatus of the plant and target specific plant genes. So in other words, the viroids have microRNAs that turn off plant genes, and that's what makes the plant sick. This is the idea. There's some support for this, but a lot of people are working on this quite actively. So silencing host genes cause disease. So in plants that have symptoms, you can see these small viroid RNAs are present in high levels, so there's a correlation between the two, although it's not yet been demonstrated that, in fact, viroid microRNAs silence any particular host gene. So that remains to be done. There's just a correlation so far. So this is a hypothesis. Otherwise, we don't know why these cause disease. They don't encode any protein. So it has to be some RNA-based mechanism. So those are viroids. Satellites are next. These are also small, single-stranded RNA molecules, slightly larger, 500 to 2,000 nucleotides. Again, they don't have genes needed for replication, just like the viroids. And they need to replicate in the presence of another virus, which is called the helper virus. So satellites require a helper. Viroids don't. They just need a host cell. But these guys usually encode a couple of proteins different from the viroids. And that is, that could be a capsid protein, or it could be some other protein that the virus needs. And the way you can propagate satellites in the laboratory is you propagate them in the presence of the helper. They can't propagate on their own. And the particles are typically very different. That's how they were originally detected in preparations of viruses. There were some other distinct particles, and those turned out to be satellites. So they encode structural protein that encapsulates the genome, again, different from viroids. And they require helper virus protein for the actual nucleic acid replication. They are not defective viruses that have evolved from the helper virus. There's no homology in the gene of the satellite with the helper. I'll give you some examples of these combinations in a moment, but I don't want you to think that they are in any way derived from that. Now, most of the ones we know about are associated with plant viruses, and they cause very different diseases than if the plant were just infected with the helper virus alone. There's a whole collection of plant viruses that we haven't talked about at all, really, with the exception of tobacco mosaic virus at the very beginning of this course. And those cause a variety of diseases in plants. And when there's a satellite present, the disease is distinct. Were you going to ask a question? No? Here are some examples. There are examples in plant viruses, bacterial viruses, and animal viruses. So there's a phage of E. coli called P4. That is a satellite that requires P2 as a helper, yes?
When both the satellite and the helper virus are inside the cell, are they both replicated, or does the satellite just use the helper virus?
They both, yeah, they both replicate. They don't interfere. Right? So we have a bacterial example, and then there are viruses within the parvovirus or parvoviridae family. Remember the canine parvovirus story we talked about last time? That virus is within this family. There are viruses in that family called adeno-associated viruses that are satellites. They require either adenovirus or herpes viruses as helpers. They don't replicate by themselves. Now, hepatitis delta virus is the best known satellite of mammals. It's a human pathogen. It's really a strange cross between a viroid and a satellite, as you will see. And the helper for this virus, which makes it a satellite, is hepatitis B virus. Remember, that's the virus with this odd gapped DNA genome with a piece of RNA in it. It encodes reverse transcriptase. So hep delta will only replicate in cells or be produced in cells that are also infected with hepatitis B virus. Now, this virus, delta, was originally isolated in Europe in cases of individuals with hep B liver disease. It was originally thought to make the disease more severe in people. It's not clear if that's any longer the case. This is now a controversial issue, whether in fact delta makes hepatitis B worse than it already is. But that's how it was originally picked up in very severe cases of hepatitis caused by hep B. So this is the current global distribution of delta. There are about 18 million people infected with this satellite. And those people all are infected with hep B. The two have to go together. Hep B can be alone, but delta has to be with hep B. There are estimated 350 million carriers of hepatitis B virus globally, and 5% of those also have delta. It was originally picked up in Europe, but with the use of vaccines, hep B has declined, and therefore delta is declining as well. But in the Asia-Pacific region, it is still highly prevalent, as you can see here on this map. And so, again, we don't know if this contributes to hepatitis severity or not, but it's certainly present in humans and replicates. So what does this satellite look like? It's a 1.7 kilobase single-stranded circular RNA, just like the viroids. It folds upon itself to form a rod-like structure, just like the viroids. It's also a ribozyme, just like the viroids, that's needed for replication. And the genome encodes a protein which is called delta antigen. And this protein is involved in encapsulation of the genome, as you'll see in a moment. When hepatitis delta comes out of a cell, it acquires the envelope of hep B. So that's the helper function provided by hepatitis B virus. It provides an envelope for delta to be in. Otherwise, it would be just naked delta RNA and it wouldn't be infectious. So in that way, it's not like a viroid. It needs a shell or a capsid surrounding it, because viroids don't need that. So the virion is consisting of the surface antigen of hepatitis B virus and the delta antigen. So the structural role for the delta antigen and the surface antigen of hep B. So here's what the genome looks like. So again, it's a circular single-stranded RNA. It is highly base paired, so it looks just like a rod in the EM, just like the viroids. So that's the genome, and this is the complement of the genome. And the genome has a site for self-cleavage. The ribozyme is right here. And there is also an editing site. If you remember, editing is the process by which nucleotides are added during RNA synthesis, which are not templated. They're extra nucleotides added. Happens in a number of animal viruses like paramyxos, and it can change the reading frame. And what it does for this virus is makes possible two different forms of the delta antigen. So there's a small and a large form of delta antigen, and they're made from one mRNA that is produced from the genome. And the mRNA has an editing site which can be used to give rise to one of the two proteins. So it's very simple, a little more complex than viroids. This genome also requires host RNA polymerase II for replication. What happens is this is a minus strand genome. It comes into cells, and RNA polymerase II initiates making a copy of this right here. And it starts to make a copy. And this red circle is the self-cleavage site, the ribozyme. And there's also a polyadenylation signal, this blue square right here. The first incomplete transcript that comes off is terminated by RNA Pol II and polyadenylated. So normally RNA Pol II is involved in the synthesis of polyadenylated RNAs. So it recognizes this polyadenylation site. It terminates mRNA synthesis here. And then there's a little bit of RNA that is continued to be made That's removed because of the ribozyme site. And then after this step, so now you have mRNA, which encodes for delta antigen. So the first transcript around the circle gives rise to an mRNA. And then the remaining ones are longer unit-length transcripts, which are then cleaved at the ribosome to give genomes. So this is now a full-length complementary plus strand RNA. It's ligated together. Again, the ribosome can seal the bases back together again. And then that's copied again to make, by a similar process, to make full-length negative strand genomes. So the copying is all done by RNA polymerase II, and the ribosome processes the concatenamers to get individual unit-length genomes. So those are the circular genomes. This is what the virion looks like. So here's hepatitis B virus, which we've looked at before. It has a genome in an icosahedral shell, and then there's an envelope with glycoproteins in it. And the delta RNA is shown here, coated with small delta antigen. So this is that rod-like RNA coated with small delta. Then the large delta is believed to mediate interactions with the envelope, perhaps with the domains of the hep B proteins that are embedded in it. So the three of the glycoproteins from hep B are embedded in this envelope. So this buds out from the cell and takes along delta with it. And probably the delta RNA is brought in there by these interactions between large delta antigen and the glycoproteins. So that's why the delta needs hep B. It needs it to supply these surface glycoproteins. Otherwise, it couldn't get out of the cell. Now, if one of these deltas would infect you without hep B, which could happen, the RNA would replicate in the cell, but no particles would be produced. So it would never spread. So it can only replicate productively in cells or in individuals that are already infected with hep B. Okay, so those are satellites, and delta is a pretty actively studied satellite because it has this association with hep B. And the last topic we'll talk about today are prions. And here we have an infectious agent, which is a pure protein, and there's no nucleic acid associated with it. And they're always in the news: bovine spongiform encephalopathy, BSE, or mad cow disease was in the news quite a bit years ago. Creutzfeldt-Jakob disease, Scrapie, Kuru, and more recently, chronic wasting disease of deer and elk. And we'll talk about these in a bit. And the fellow who created this name, prions, and made the whole hypothesis that it was an infectious protein was Stanley Prusiner, who got the Nobel Prize for that in 1997. These prions cause what we call transmissible spongiform encephalopathies, or TSEs. These are infectious agents without genomes. In a variety of mammals, they cause fatal neurodegenerative diseases. Okay, so an encephalopathy is a disease of the brain. That's what these prions are causing. Thousands of people are diagnosed globally with these each year, about 1% of which arise by an infectious route. We'll go through the different ways you can get a spongiform encephalopathy, but a fraction are the infectious kind. By 2002, 120 humans had contracted Creutzfeldt-Jakob from eating meat of animals that had BSE, which is also a prion disease. We'll talk about how that happened in a moment. And of course, encephalopathy, again, disease of the brain. These are the various TSE diseases of animals. These are prion diseases. So we have BSE. We have CWD, which infects deer, elk, and moose. We have exotic ungulate encephalopathy, which is a disease of Nyala and greater kudu. The feline spongiform encephalopathy, domestic and great cats. So these are cats in zoos or cats in your house. Scrapie in sheep and goats, and transmissible mink encephalopathy. All right, those are the animal ones. And then there are human TSEs: Creutzfeldt-Jakob, fatal familial insomnia, Gerstmann-Straussler syndrome, Kuru, and variant CJD. We'll talk about some of these subsequently. So Scrapie was the first transmissible spongiform encephalopathy that was recognized. It was a disease which caused infected sheep to have a neurodegenerative disease, and part of that was they would rub themselves on fences. So the farmers noticed this. They would rub their fur off, and the farmers called it scrapie. And the French call it tremblant du mouton because these animals also have uncontrollable trembling. They have motor disturbances. They waste away and eventually die within a couple of months. So that was called scrapie. It was recognized over 250 years ago in European sheep. So as far as we can tell, this is a natural disease that arose in sheep. And you'll see the genetic basis of it in a bit. In some countries, it's endemic. 1% of the sheep in the UK, for example, every year come down with scrapie still. There's no way to prevent it at the moment. So that's a sheep with scrapie, and this poor guy has scraped off a lot of his fur by rubbing up against the fence. And again, it's a fatal disease, always fatal in sheep. So a very long time ago, farmers observed that infected sheep would transmit this disease to others. So if they had healthy herds and diseased sheep, eventually the healthy herds would become infected through contact with the other sheep. So they said, this must be an infectious agent. And in 1939, it was shown that if you took a sheep brain from an animal with this disease, make a homogenate, and pass it through filters that would pass only a virus, that homogenate, that filtrate, could cause disease in other sheep. So it looked like this might be a virus, and people started to study this and try and understand what kind of a virus it was. But it turned out to be extremely resistant to all kinds of things that would inactivate viruses, including ultraviolet light, ionizing radiation, formaldehyde, even autoclaving and boiling. The infectivity of these preparations withstood all those treatments. Now, you can never, in fact, get rid of all the infectivity in these preparations. You can boil them or autoclave them. You get rid of a lot of it, but not all of it. And at some point, it was proposed, and Prusiner was the main proponent of this theory, that these agents didn't have any nucleic acid in them. They were simply too resistant to all these inactivating agents, and there was no nucleic acid detectable. So they were clearly atypical infectious agents. And this is just an example of some of the early experiments that were done. And this is a graph of the size of the genome of the virus versus the amount of ultraviolet radiation. Sorry, not ultraviolet. This is radiation dosage from less to more here. And you can see there are a bunch of viruses, both DNA viruses and RNA viruses. So the largest viruses are inactivated rather quickly with low doses of UV because their genome is big, it's a big target. And as the genomes get smaller, it takes more and more radiation to inactivate it. So this is a measure of how much radiation you need to inactivate infectivity. And so the DNA viruses fall along this line pretty much, and the RNA viruses fall down here. And there's a correlation between the size of the genome and how much radioactivity. So the scrapie agent is way down here. It takes more radiation than almost any virus except porcine circovirus, which we've mentioned a few times, these viruses with single-stranded DNA genomes. So it was very unusual in its resistance to irradiation. Now, spongiform, that's just part of the names, transmissible spongiform encephalopathy, arose because the brain had holes through it. It looked like a sponge. And we'll see a picture of that in a moment. All of these encephalopathies have psychomotor dysfunction in the infected animal, and they are all slightly different. Depending on the animal, different parts of the brains are infected, and the diseases are slightly different as a consequence. So each disease, whether it be in cows or sheep, has a characteristic pathology. So here's an example of the spongiform encephalopathy. This is a section of a sheep with scrapie, and you can see the distinct holes in the brain matter. Now, all these TSEs that we've mentioned so far, the animal ones and the human ones, they have very similar histochemical abnormalities, which have been studied very extensively. They're all listed here. Plasma membrane defects, vacuolization, loss of neurons, this spongy appearance, clumps of glial fibrillary acidic protein occur, which is not normal. And finally, the formation of fibrils in the brain, which are called amyloid. And these are fibrils made of amyloid precursor protein. In early studies, the agent can be tracked by injecting it into a susceptible species. So you have a sheep with scrapie, you can make a brain homogenate. If you have sheep, you can inject it into sheep. But more recently, people have been injecting these homogenates into mice and other animals to try and study the disease. And they develop the disease. And you get these kinds of symptoms: difficulty walking, dementia, and death. Now, the problem with this research is it takes a long time. Sometimes the incubation period is a year. You infect your hamsters or mice, and you have to wait a year for the disease to take place. So they're really heroic experiments. When you inject these infectious extracts of brain into animals, they accumulate in the lymph tissues initially, and then they spread to the CNS. Okay? So however the route of entry into the host occurs, the lymph tissues appear to concentrate them initially. And then in the central nervous system, where the protein begins to do its work, as we'll see, you have the typical spongiform pattern and the loss of neurons. There's no inflammatory response. So very differently from many of the virus infections we've talked about, you don't have an innate response. You don't have cytokines produced. There's no fever, soreness, muscle ache, etc. No antibody produced. There's no T cells. And that was an early clue about the nature of this disease. The problem with these, they're not very common, all right, but you cannot detect them before you have symptoms. And by the time you have symptoms, you're well on the way to dying. There are no diagnostic tools available at the moment. The only way we can detect infection in an animal is to take the brain out and do a Western blot. So there's no way to do that in people, obviously. Now, people are working on diagnostic tools, but the only way to do it right now is by antibody reagents. So we don't know who's at risk. We don't know who's carrying this infection. There's no way to treat it. So you come down with Creutzfeldt-Jakob. There's nothing we can do for you, and it's uniformly fatal. And you can't even alleviate the symptoms. The disease is caused by proteins. And in fact, the mathematician way back in 1967 who knew about scrapie said it could be that they're protein only based on purely theoretical analyses of the disease patterns. Now, Prusner in 1991 purified a protein from infected sheep brain and showed that this could be transmitted to other animals. So he showed that the protein alone was sufficient to transmit the disease. And he got tons of flack for years and years. No one believed him. They said, well, you have some nucleic acid contaminating your protein. It just can't be that a protein could be infectious. But he persevered, and he seems to be right. He called it a prion, an infectious protein. He also sequenced the protein and found out the gene that encodes it. It is called the PRNP, prion protein gene. And this gene is absolutely needed to develop these TSEs. Okay? What we think now, the current view, is that the pathogenic protein is a conformational isoform of a normal host protein. So the normal host, we all have the PRNP gene. We make a protein, which is called PRPC for cellular. It's a normal protein. It's not pathogenic. We need it for a variety of neurological functions. And something goes wrong, and it folds aberrantly, and it becomes pathogenic. The cellular protein is largely on the surface of neurons. It's got an unusual anchor. It's GPI linked. It doesn't have a transmembrane sequence, a glycophosphatidylainositol anchor to the membrane. The abnormal version of this protein, when you put in a host, causes the normal protein to be converted to the pathogenic conformation. So you have an animal, a sheep with scrapie. You purify prion from that animal. It's called PRPSC for scrapie. You inject that into a healthy animal. That protein causes the normal PRPC to be converted to the pathogenic protein. So it's an abnormal conversion. And what happens in disease, this PRPSC, which is highly resistant to proteases, and it aggregates, it accumulates in the brain, and that's what makes the disease. It causes the fibrils to accumulate, and it makes what are called plaques. So here's a typical staining of an animal, a section from the brain of an animal with a prion disease. So this animal was injected with an extract of brain containing prion, and it's stained with antibody to the prion protein. You can see they form these very typical plaques, which are basically long tangles of protein. The prion protein forms these fibrils, which are called amyloids. In my view, one of the best experiments was when mice were made that were knocked out for the PRNP gene. And these mice survived, so the gene was not essential. But then when you tried to infect these mice with pathogenic proteins, they're completely resistant to infection. Right? So the normal gene is essential, and that makes sense because the host has to make the normal protein in order for it to be converted to the pathogenic protein by PRPSC. Now, if you take these knockout mice and you add a transgene, you now make them susceptible to infection with a protein extract again. Okay, so really important experiment. The prion, the pathogenic version, PRPSC, can be introduced into a host by infection. So we can give the protein to an animal in the lab, or in nature, an animal can acquire it from another animal. So in all the years that sheep passed prions from one to the other, that was a natural infection. Somehow the sheep were passing prions to an uninfected animal. So that's the infectious kind. Those are the transmissible TSEs. But you can also have rare mutations that arise in your gene that will spontaneously cause it to misfold, and you will develop a spongiform encephalopathy. It's no longer transmissible. You haven't acquired it by infection, but it's the same basis for the disease. The ideal proof of the whole hypothesis would be to make the pathogenic protein in vitro and infect an animal and show that the animal gets scrapey. But that's not easy to do. There is no sequence difference between the pathogenic protein and the normal protein. It's just a folding difference. So you can't simply make the pathogenic protein in vitro and give it to an animal, because when you make it, it folds normally in vitro and it doesn't cause disease. The only way that you can make an abnormally folded protein in vitro is to add some abnormally folded protein to the normal protein. And that has turned out to be very difficult. The conversion is not very efficient, and when you infect animals, you can never tell that it wasn't the original misfolded prion in that prep that caused the disease. Okay, you see the conundrum there. So we don't have the final linkage here, but all the genetics really supports the idea that the prion protein is the essential agent and misfolding is involved. The cellular version of the protein is shown here in a linear view. It has a lot of alpha helical content. The structure of this protein has actually been solved. It's a well-behaved protein. As I said, it's linked to the membrane by a GPI linkage. That's there at the C-terminus. If you treat this protein with a protease called proteinase K, it's completely degraded. Right? Now, the scrapey version, or the pathogenic version of the protein, is resistant to protease digestion in vitro. The protease only clips off a little bit of the N-terminus. So this is, in fact, the assay that is used when you're infecting animals with scrapey, with the pathogenic version, and you want to see if they're infected. You purify the protein, you treat it with proteinase K to get rid of all the normal protein, and then you do a Western blot. So whatever appears on the Western blot using antibodies to PRP is going to be the pathogenic version, because the normal cellular version will have been degraded. So you can't make antibodies specific to the pathogenic version. No one's been able to do that. It's the same protein sequence, and the conformational differences obviously haven't been picked up yet by antibodies. So the conversion goes from this nicely behaved alpha helical protein to a protein that has a lot of beta sheet in it, and that's shown by this drawing here. We don't have the structure of this protein because it aggregates and behaves very badly, and all we can say is that it's resistant to digestion and has a lot of beta sheet structure. But that's the pathogenic protein, that's the normal protein. This causes encephalopathy, this doesn't. Same amino acid sequence, that's the key here, just some structural difference. So how does this happen? How does a pathogenic prion convert a normal protein? Remember, that's what's going on here. You purify the abnormal protein from sheep, you inject it into new sheep, And that abnormal protein converts the sheep's normal prions into abnormal ones. How does that happen? Well, these are two of the hypotheses shown here. The first one is called the refolding model. The idea is that no conversion between the cellular form and the scrapie form or the pathogenic form of the two proteins normally. But if some scrapie version is introduced, it will then drive the normal protein to change its conformation and become altered and pathogenic. And then the more you make of these, of course, those can go back and continue to convert all the normal cellular proteins. Now, we don't know how this happens. I have no idea. All we know is that you mix the two proteins together and the pathogenic one alters the conformation of the normal one. The other model says that in us there is a normal conversion between normal and pathogenic proteins. It's very, very little that occurs. But when you have enough of this present, which happens in a variety of conditions, it will then start to form aggregates, which will then proceed quicker and quicker through this conversion process. At the moment, we don't know which one of these is correct. So how do you get these infectious proteins into your brains? Well, in humans, they're often acquired by ingestion. There are other routes as well. So the idea is that when you ingest it, here's your intestine here. It's taken up by an M cell in a Peyer's patch, goes to maybe dendritic cells and lymph nodes, and eventually reaches the brain and spinal cords either through the blood or perhaps through peripheral nerves that bring it there by axonal pathways. We do know that when the proteins are injected into susceptible animals, they do accumulate in lymph nodes and dendritic cells, so they may facilitate the uptake of the pathogenic protein. This is something that isn't really well understood. So let's talk about some human TSEs. The first one that was ever discovered, so remember sheep was the first TSE discovered, that's in an animal. The first human TSE was discovered in New Guinea. It was called Kuru, and that was an encephalopathy in the 4 people of this country. It had a 30-year incubation period. So this was a disease that was known to epidemiologists, but no one understood what was causing it. Carlton Gajdusek, who was a scientist, went to New Guinea and studied it for many years, and he concluded that it was spread by cannibalism. So someone would die, and they would probably have a transmissible spongiform encephalopathy. How they got it, we'll talk about a bit later. And the tradition in this, among the 4 people, was that the men get to eat the muscles of the dead person, and the women and children eat the brain. And this disease was mainly found in females and children, so this is consistent with the cannibalism. So he told them you should stop doing it, and they did, and the disease went away. So the idea has sustained that this was transmitted from person to person by eating the infected brains. So he told them to stop, they listened to him, and Kuru stopped. He eventually got a Nobel Prize for figuring this out. So human TSE, some of them are infectious or transmissible. So if you eat, yeah, I'm sorry, yes?
So is it understood how the proteins aren't broken down in the gut? How the prions aren't broken down?
Remember, the pathogenic, how are the prions not broken down in the gut? The pathogenic versions are very stable to proteolytic digestion. Only that N-terminus is taken off, and the rest is no problem. It's a very, very hardy protein. Yeah. So there are infectious or transmissible spongiform encephalopathies. You eat protein, or you're contaminated in some way with the pathogenic protein, and you get the disease. So Kuru is one of those. You eat the brain of a victim of SE. More recently, these diseases have been spread by transplantation of corneas from people with Creutzfeldt-Jakob. You don't know they have Creutzfeldt-Jakob very early in the disease. There are pathogenic prions present, but they have no symptoms, and they die for some other reason. They take their corneas and put them into someone else, and they get Creutzfeldt-Jakob. Hormones purified from the blood, same thing. Transfusions, these have all been shown to transmit Creutzfeldt-Jakob, which is a transmissible spongiform encephalopathy, to people. Then we have BSE. Bovine spongiform encephalopathy is another infectious one. The cattle acquired it in the UK because they were fed feed containing protein, which was ground up animals who had died, and among those animals were sheep that had scrapie. Okay, now, cows are vegetarians normally. They feed on grass. But if you are raising a cow for meat, you want it to grow really quickly, so you want to give it protein. Here in the U.S., we feed cows soy protein because it's cheap and we have a lot of it. But it's not easily available in Europe, so they feed ground up other animals to the cows to give them protein. So this made the cows get spongiform encephalopathy in the 80s, and then it got transferred to people who ate the cow meat. So that resulted in what's called variant CJD, and this is different from the Creutzfeldt-Jakob that we normally see. Had a different incubation period, slightly different pathology. You eat BSE contaminated beef and you get variant CJD. And this is a form of forced cannibalism, just like the kuru. You're forcing the cows to eat meat which is infected, which contains pathogenic prions. So that's the epidemic spread, and this was an epidemic. Hundreds of thousands of cows were infected in the UK, and millions were slaughtered to get rid of this disease by feeding them contaminated meat, processed animal byproducts. And this was stopped. So at one point it was realized, hey, we're doing something wrong here. And so they said, we're going to stop byproduct feeding, and the transmission to cows stopped. But the incubation period is many, many years. So it took a long time for this to pass through. And eventually, well, they did slaughter most of the cows in the UK, and it took a long time to pass through the human population. So I believe that consumption of BSE-infected beef gave people TSEs. A lot of people don't believe this, but in fact everything is consistent with that. And again, that's variant CJD. It's not the same as Creutzfeldt-Jakob that we see in humans. Here's a graph of the cases of BSE and variant CJ. So the BSE in cows, these are thousands of cows. Starting in the late 80s, there was an increase in BSE in cows, and this can be recognized. The cows couldn't stand up, they have neurological deficits, they waste away, and it was discovered as a prion disease. They stopped feeding the cows meat and bone meal, ground up meat and bone meal, and the epidemic continued, and then eventually in the 90s it started to go down, and then there was a ban on mechanically recovered meat, which means you take carcasses of animals and you pump water or air through them at high pressure to recover the meat from the bones, and in doing so you release a lot of the spinal material into it. And so eventually the BSE outbreak in the UK went away. There's still a low level in Europe, but you can see the variant CJD in people overlapped that. It peaked and that now has gone down. It took a long time for the incubation period to get through all of this. So this is getting a transmissible spongiform encephalopathy by eating infected cows. Now there's another kind of Creutzfeldt-Jakob called sporadic. It's not an infectious kind. Somewhat less than 1 million people worldwide. Typically they're older, and it accounts for about 65% of the spongiform encephalopathies. And this happens with no warning. You start to not be able to walk properly, and you get worse and worse, and eventually you die of CJD. Maybe the most famous victim was George Balanchine, who was the head of the New York City Ballet. And you have normal prion genes. Right? There's nothing wrong with your prion gene. And if you don't know that you have this, you could transmit it to others. So if you give blood or a cornea, you will transmit it to others. So it goes from a sporadic kind of disease to infectious kind. And we think that maybe Kuru was established originally years ago in New Guinea by someone who had sporadic Creutzfeldt-Jakob. They just developed it, and that person died and was eaten, and that started to spread the disease among the tribe. So remember, what's happening here is you have a normal protein. It starts to misfold for reasons we don't understand. And as soon as there's a little bit of misfolded protein, it catalyzes even more and more misfolding. So you develop the disease. You have no infection to account for that. There's no mutation. There's some kinds of CJD which are associated with specific mutations in the gene. That's not the case here, and that's why it's called sporadic. There's also familial, which is associated with mutations. This is an inherited disease, so you have a mutation in your prion gene, and that predisposes it to misfolding and development of Creutzfeldt-Jakob. It's associated with an autosomal dominant mutation in that gene. And again, if you have this mutation and you're starting to develop misfolded prions and you give products to others, you will transmit it to them. And this is how often CJD was transmitted to others via these products. So we have three types, basically, of spongiform encephalopathies. We have infectious or transmissible, where a sheep transmits it to a sheep, or we get it by eating contaminated beef. There's familial, where a mutation is involved, and specific amino acid changes in the prion gene have been identified that are responsible for this. And then we have the sporadic, which just happens. We start to misfold our prion, and we don't know really why that happens. All three diseases can be transmitted experimentally. So whether an animal has infectious, familial, or sporadic SE, you can make an extract of the brain and transmit it. So once you have a misfolded protein, it's infectious. It can be transmitted to others. So here in familial and sporadic, they appear spontaneously without an infectious component, but then you can transmit them to others because the misfolded protein is infectious. That's the whole basis for this prion hypothesis. So here's again another summary of this because I think this is really important. Here's the normal prion protein, nicely folded alpha helices. If you mix it with a pathogenic version, if you eat contaminated beef, it converts this to the scrapie form, and you get a spongiform encephalopathy. And then your prions are now infectious, or the prions of an animal that have acquired it this way. And remember, the protein misfolds, so it has lots of beta sheets. There are familial forms where you have mutations in the prion protein that predispose them to misfolding. So as you age, it misfolds, and you develop the disease. Those are the genetic disease. And then we have the spontaneous forms where the proteins just spontaneously misfold. There's no mutation, there's no infectious component, and we call those sporadic. But in the end, they all produce this misfolded prion, and that is infectious, so can be transmitted to others. Okay? Yeah?
How dangerous is it to work with prions in the lab?
You have to be careful. You have to wear gloves and proper protection. It's not BSL-3. The most effective way to acquire them is to ingest them. But if you have cuts and you put it on, it would get into your blood, so you have to be very careful with them. But the cross-species transmission is very inefficient. If you're working with human prions, then it's a problem. But often the labs work with animal prions, in which case going across to other species is much less efficient.
One of the interesting things about the familial diseases is that as they pass from generation to generation, the onset becomes earlier.
Do we know why?
No.
So there are no additional mutations that are sustained in the gene. The species barrier is an interesting one. So remember, you take an animal with prion disease and you make a brain extract, you put it in another animal, that animal will get the disease. But it turns out that if you go into different species, the disease is inefficient, as we were just saying. And for the most efficient transmission, the sequence of the protein should be isologous, should be the same sequence in the donor and the host. So for example, mice, if you infect mice with an extract of cow brain, and a cow that died of BSE, it's very inefficiently transmitted to the mouse. But if you give the mice the gene for the bovine form of the prion, you make a transgenic mouse for the prion gene of cows, and then you infect the mice with cow prions, then the transmission is very efficient. So something about templating the misfolding requires that the proteins be the same sequence. So the sequence of the protein is a barrier to interspecies transmission. It's not an absolute barrier. It does happen. It's just very, very inefficient. Now, the interesting thing with respect to this barrier is that the BSE prion has a very broad host range. You can infect many different animals with this protein and reasonably efficiently. So this is different from most of the other prions in that infects many animals, and that's probably why it went into cows originally and why it went into people as well. So that's the reason why it's scary. And some prions can overcome the primary sequence limitation that I just talked to you, and we don't understand that. And that's why BSE is scary, because it can infect a lot of animals. Interestingly, there are also strains of scrapie. So if you put scrapie in different kinds of animal hosts, they develop characteristics, incubation time, glycosylation, profiles on gels, and these are called strains of scrapie protein. These are not sequence differences. These are protein conformational differences. And so that just the conformation can differ enough to say, change the incubation time in one animal versus another. Very recently, it's been appreciated that these proteins can evolve. All right? They can actually undergo Darwinian evolution. So if you can take a prion from an infected animal that has prion disease and put it into a cell culture, and it will enter the cell and template the endogenous prion to misfold in that cell. And then as you split the cells and passage them, they will maintain the misfolded protein. It doesn't hurt the cells because they're just cells. They don't have a nervous system that gets affected by the misfolding. And when you do that, the prions get heterogeneous. You see strains of cells. So they're undergoing mutation in the cells, even though they don't have nucleic acid. So, for example, if you take brain prions and put them in cells, they become adapted to the cells. And then if you could do competition experiments with the cell-adapted versus brain prions, putting them in cells at the same time, the cell-adapted ones do better. So there are clearly differences that occur when you pass these in cultured cell. There's a drug called swainsinine, which is an inhibitor of glycosylation. And if you put prions into cells and treat them with this drug, you inhibit the conformational change. But after a few cell divisions, prions return that are completely resistant to the inhibitory effect of this drug. So look at it another way. This drug inhibits the conformational induction of change caused by the pathogenic protein, but some variants arise that can resist the drug. And those don't have any differences in the amino acid sequence. They're just folding variants, slightly different structure. I think this is amazing, and it shows that they can mutate and be selectively amplified. Just like a nucleic acid can mutate, we could select out drug-resistant variants in a viral genome. You can do the same with a protein, apparently. There is a disease called chronic wasting disease, which affects deer, elk, and moose. It's the only one affecting free-ranging wildlife. All the other encephalopathies affect farm animals or animals in zoos. So this one has got out, and that's why this is a concern, because it's spreading across the U.S. and Canada. These are areas of the U.S. with chronic wasting disease, and there's some in New York and Virginia. And here, these are deer, just deer walking around, and they develop a spongiform disease, and eventually they die. And of course, it's an issue if you're a hunter and you like to go out and hunt deer, because you could eat a deer that has a spongiform encephalopathy. So there's a lot of concern about this, and there's a website called Chronic Wasting Disease Alliance, which is absolutely geared towards hunters to tell you, you know, what to do if you see a weird deer and how to dress it and so forth. So this is a concern because maybe it can come back into people. We don't know how it got into deer, but it may have spread from a cow with BSE originally into the deer population. So here are some scenarios for how these TSEs have spread over the years. So the blue color is naturally occurring disease. So we had scrapie all the time in sheep. It was probably always there, probably arose spontaneously in some sheep years ago and then was transmitted by infectious processes. This could have led to infection of deer and elk in the wild, contact between sheep and deer and elk. And so those are thought to be natural diseases. So the scrapie led to contamination of cow because the ground up sheep were fed to cows, so that we have BSE. The BSE was eaten by people, so now we have variant Creutzfeldt-Jakob, and then they transmit it to other people by transplants and blood products and so forth. We also have sporadic CJ in people, which is a natural disease. also transmitted to others by contaminated transplant material. Then all of these diseases in cattle, various animals, lions, tigers, primates, these are all zoo animals, and your cat, all happen most likely from feeding them cow products, products from an infected cow. So the cat food used to have ground up cow in it, and they feed animals in the zoo pieces of cow, and so that infected all of these animals. So it began as a natural disease, but by our manipulations, we've spread it even more. So it's sort of an extension on the emerging virus theme that we talked about years ago. So we're worried about prions in the food supply. We don't really understand this disease very well, so there are a lot of efforts aimed at protecting it and finding ways to prevent the disease, because they're there. The prions are in the food supply, and so when this disease arises, we have to be able to deal with it. We've got some diagnostic tests that have been developed for that, and companies are screening for drugs that will inhibit prion accumulation. Now let me end with just leave you with this interesting thought that not all prions cause disease. Similar proteins have been identified in yeast and fungi, and they don't cause disease. They actually create protein-based memories. And what these proteins do, because they can switch between one folded state and another, they have different phenotypes. Not a disease phenotype, as I've told you, but they just have a different phenotype that allows the organism to adapt to a different environment. And since the switching happens very quickly between the normal and the misfolded state, an organism can respond rapidly to, say, a change in the environment. Okay, so this is thought in the case of yeast to allow them to really quickly adapt to stresses. And so here are some of the prions that have been identified. So here's the one we've been talking about today, the mammalian prion that causes disease. But these others are all genes in yeast and fungi that have the same properties. There's one version and a misfolded version of the protein. And the misfolded protein has very different properties. So let me just show you one. There is a gene in yeast called URE2. So the function normally is to repress transcription of genes that are used to break down nitrogen. But in its misfolded state, the prion state, it allows indiscriminate utilization of nitrogen sources. So that's not a bad thing. If the yeast is stressed and it needs nitrogen, this misfolding can occur very quickly, and the yeast can use whatever nitrogen source is available. Then when things return to normal, the protein goes back. So that's the idea about these normal proteins, and people are looking for them in our genomes now as well, because it may be that the abnormal prion that causes disease is actually a version of, is related to other proteins, or is functionally similar to other proteins that allow us to adapt to stressful environments. They're very cool things.